I'm Andrea Trese, and this summer I participated in engineering research through the University of Michigan's Environmental Water Resources program. And I'm with Professor Hayes and Professor Raskin. I've been involved in a project regarding the removal of arsenic and nitrate from drinking water sources. The reason this problem is really important is that it has negative long-term health effects. So chronic exposure in drinking water can cause a variety of diseases from arsenic poisoning, including cancer, stroke, diabetes, heart disease. As you can see, this woman in the right, and her hand is covered in black warts, and this is because she's had drinking water contaminated with arsenic for a long period of time. In fact, 1 in 5 deaths in Bangladesh can be linked to arsenic, according to a recent study. The World Health Organization recommends a limit of 10 parts per billion, which is the same as the US EPA drinking water standard. Arsenic is naturally found in groundwater, and it's found in especially high concentrations in Nepal, India, and Bangladesh. But as you can see from this map, it's also found in other parts of the world, including the United States and Canada, so it's also a problem in developing countries. Another part of the issue is that the current method for arsenic removal is unsustainable. It involves the precipitation of iron hydroxides, which are oxidized. And so when these solid wastes are put into a landfill, the landfill, which is in a reducing environment, reduces the solids, and that releases the arsenic. And in that way, the arsenic can reappear in water sources. So the solution being researched is a biologically active carbon reactor system, which is capable of removing arsenic and nitrate. The system works by using microbial communities to provide sequential removal of the contaminants. As you can see in the figure on the right, the colors represent different microbial communities which perform different steps of the arsenic removal process. The microbes reduce contaminants, enabling them to form precipitates. And in this case, we use iron sulfides to remove arsenic. And these reduced iron sulfides are our way for removing arsenic and leaving clean drinking water. In this graph, you can see how water comes in at an influent concentration of over 300 micrograms per liter, and as it travels through the reactor, the concentration is reduced, and it leaves with an effluent concentration of less than 50 micrograms per liter. In the same way, nitrate concentrations and sulfate concentrations are also reduced as water goes through the reactor. Some advantages of this system include that it simultaneously removes 2 contaminants that are naturally both found in groundwater, and that saves on cost and energy. And we also expect that it would prevent the release of arsenic in landfills. Because the iron sulfides in our system are already reduced, we expect that they would be stable in landfill conditions. My experience this summer has been really engaging. I got to do some hands-on work with the reactor. I got to conduct some phosphate batch experiments, and I really learned a lot by helping with RNA experiments and solids analysis. The future of the project includes plans to evaluate post-treatment of effluent and stability of waste solids, to finish the process of acquiring a patent, and to conduct a feasibility study in India or Nepal in collaboration with the Institute for Social Research. We also hope to explore practical and challenges and cultural factors regarding the use of this system in South Asian countries. So I want to give a special thank you to Tara, Mark, Girdar, Professor Raskin, and Professor Hayes for a great summer research experience.